Undergraduates at the University of Wisconsin-Madison are fortunate to be presented with the opportunity to work with some of the leading researchers in a range of fields. Located in the Wiseman Center on campus, Dr. Chang's lab is one of the many research labs which create an extensive community based around discovery.
I think I was interested in doing research from earlier times, probably in college. In the field of study, I wasn't really sure. You can see that I got a bachelor's in biochemistry and molecular biology, and when I did my PhD, it was in neuroscience. And then, I guess, at that time, I was interested in how the brain works. And the reason I decided to go to biology in college was I wanted to either study something small or something big. Something big would be astrophysics, something small would be molecule. And then what drew me to life science was because there's life. And I still remember in graduate school, there's one day I looked at a live neuron under the microscope at 100x, and I can see the cell literally breathe. I guess I could call myself a neuroscientist, developmental neuroscientist. What makes my research unique is that I use genetic tools. So genetics would allow you to manipulate genes in the genome at your will, essentially. Take it away, put it back in, regulate its expression, turn it on, turn it off. That would allow you to assess function. In general, we're interested in DNA methylation-dependent epigenetic regulation of brain functions. The focus is on central regulator of the process. It's a methyl CpG binding protein 2. Basically what it does, it's binding to methylated CpGs in the genome. It would recruit transcription regulators, chromatin remodeling factors to the site to either change transcription profiling or change the chromatin structure. If you have mutation in this important gene, then you would have a severe neurodevelopmental disorder called Rett syndrome. There's many symptoms. So we would need to understand the cellular and molecular mechanisms underlying the steps between the mutation and the disease.
Rett syndrome is a genetic disorder which exclusively affects females. Roughly 1 in every 10,000 girls will develop the condition and will begin to show signs by about 6 months old. Rett syndrome causes social anxiety, withdrawal and disinterest in people, difficulties with muscular development and coordination, problems breathing, and seizures. While most children with Rett syndrome share a specific mutation in the X chromosome, it is currently unknown what the gene does or how the mutation leads to Rett syndrome.
And the hope is that the more we understand about the mechanism, the better we could design treatments or strategies to help the patients. The lab has two separate areas that are interconnected. One is basic research on the function of the gene. The other one is disease mechanism and therapy development. The two most important things I view is interest and commitment. You're interested now, but in future you lose the interest, then you would not be able to sustain your career in this area. So interest is number one, you have to have it, and then you have to cultivate the interest and you have to maintain the interest. The second one I think is going to be a commitment. How much are you willing to put into it, your time, your effort, your resource, to keep moving forward? We have developed many tools as a field to manipulate genes in the genome. So how to use these tools to improve human health, I think that is the question. We can already show in cell models, in animal models, that you can correct mutations in disease-causing genes and cure the disease. But we haven't been able to do that in humans. Right? So I think that is going to be the future. It's going to be you guys who will maybe develop new tools or maybe develop those checks and balances so that they can be used safely to improve human health.